Very good evening to all, and thank you so much for an excellent presentation, sir. So I would be touching on to the basic, because before we start, let us be clear about what is this SARS-CoV-2 virus, its structure, and what are these mutations which are occurring on and on, because of which the COVID is still continuing. And I bring greetings to you from our Institute of Liver and Biliary Sciences, which has a dedicated clinical virology setup. Oh, next slide, please. Yeah. So the coronaviruses are not new. In fact, they were, although 1960 onwards, we started seeing these coronaviruses, but they were the common, very causing mild upper respiratory tract infections in the form of coronavirus 229E. There were only 4 pathogenic strains of the coronaviruses to humans. Then in 2002, they became slightly more severe. They caused severe first type of SARS, that is a severe acute respiratory syndrome coronavirus, which caused pandemic. And then following that, there was another pandemic, MERS-CoV. But these were curtailed within a couple of years. 2019, everybody saw towards the end of 2019, the virus took a huge mutated form, and it caused pandemic. It more than 700 million people got involved globally. All the countries got involved, and it's still continuing and growing on. Next slide. So how this virus came? So all the coronaviruses actually came from the animal. These are driven from the zoonotic species. So they always have a natural reservoir, and mostly it is the bats, few in the rats, but it is mostly the bat. Now, till 2002 and 3, it was just causing very mild kind of an outbreak. But following that, the changes occurred so much in this coronavirus, which originated from these bats, that it led to a massive pandemic, and also the lower respiratory tract infections, pneumonia, all those things occurred. Now, interestingly, this COVID-19 or SARS-CoV-2 virus showed close relation with a particular type of bat, more than 96% resemblance, which we call as rhinolophus bat, RATG13. But why I have put a query that the intermediate host is not known or its origin is still questionable is that few regions of this virus resembled very closely to this bat virus. But there were few stretches inside the virus where it did not show much of the relatedness. I'll show you that region in my subsequent slides. So its intermediate host is still a query, and that has caused a huge pandemic, which is still going on. It showed less similarity with the previously existing SARS-CoV coronavirus group, SARS-CoV-1 and MERS-CoV. Next slide, please. So this virus has actually evolved from the bat, and it came to us. Talking about the structure, it is a single standard positive sense RNA virus. This is how we classify the viruses: RNA, DNA, single standard, double standard. It belongs to the family beta coronavirus and sarbecovirus subgenus. There are the virus is a huge, it's a longer structure. So you can see it has got about 30 kb, 30,000 base pairs are there into it. Important are the 4 structural proteins which undergoes changes, and those are responsible for all the pathogenicity and the virulence. Spike protein. Now, the word corona is derived from these spikes which are protruding out, giving it a shape of a crown-like structure, and that is why the spike protein is also very, very important, which is required by the virus for its entry into the cell, and this is the one which is most susceptible to all the mutations and all the changes which are happening. Then the other 3 important proteins are membrane protein, envelope protein, and the nucleocapsid protein. Next slide. Spike protein, I really want to discuss here in detail because this is the most important protein and how it has evolved from how it has become so pathogenic from the previous coronaviruses. This spike protein, as you can see, consists of two subunits, S1, S2, which a small region of amino acid, four base pairs, four amino acid region joins them, as you can clearly see. Now, SARS-CoV-2, you can see in this region differs from all other bat coronaviruses as well as the previously existing pangolin and other to which it showed quite a resemblance. Now, there was a four polybasic sequences, PRRA, which was inserted at the site. Now, why this is important? Because this gave an advantage to this COVID-19 that this breakup of SARS S1, S2 unit is faster in it as compared to the previous coronaviruses. Now, once this cleavage is better, more pathogenicity, faster processing of the virus is done. And that is why the evolution of this COVID-2, SARS-CoV-2, COVID-19 from the previous SARS viruses is that it is more virulent, more pathogenic because it has that unique amino acid sequence at the cleavage site of S1 and S2. So its processing or internalization is much faster as compared to the previous coronaviruses. There is another interesting unit on this S1 subunit, which we call as receptor binding domain. Now, this is the domain of the spike protein to which it binds to the receptors. Now, when the mutations happen to these two regions, either the receptor binding domain or this cleavage size, all adds on to the pathogenicity, virulence of the virus over which it has evolved over the years. So the next slide. Now, once we talk about how this virus replicates, so the infection mode of acquisition is through the nasopharynx. The spike protein, which I said, with the same S1, S2 unit, goes and binds to the receptor. These receptors are present in the nasopharynx and they are present at other places also. These are known as ACE2 receptors to which it binds. Now, the cleavage of S1, S2 occurs. Now, for this cleavage, even the host tissue proteases are required, which are in the form of TMPRSS2 and furin receptors, which adds on to its internalization, faster processing, and then the virus replicates and releases. There are four important proteins I've again and again said. Spike, I told you, binds to ACE receptor, helps in internalization. S1, S2 cleavage is important for virulence and pathogenicity. The envelope protein, E protein, allows the virus to assemble. Now, another important thing about this E protein is that it also forms the ion channels or the viroporins through which the virus can modulate the host cell environment. Now, any mutation occurring in this will have an immense inflammatory response inside the body or will lead to a lot of inflammasomes production. So this protein is important from that point of view. M protein allows the virus assembly and the N protein helps in the RNA replication. So mutations in the N protein also will help in faster processing, faster RNA replication and the adds on to the pathogenicity. Next slide. Now, how the variant? So there is a parent virus. Now, anything changes with that occurs. Now, RNA viruses, they are prone to mutations. First, the copying mechanism means RNA polymerase forms a copy of it. Sometimes this is defective, so there is a lot of errors are created. Then you have a mechanism by which this copy which is generated during the replication is checked, which is known as proofreading. So certain viruses have an error-prone proofreading. If these proofs are not read properly, again, the changes occur in the genetic structure. Now, these genetic changes are known as mutations. Next slide. And if the changes in the genetic structure makes the virus change in its physical structure, pathogenicity, clinical manifestation, then it becomes a variant. What is there in the SARS-CoV-2 which has added on to the genetic diversity? As I had already showed you, it has a very large genome, 30 kb, 30,000 base pairs. So longer the genome, more are the chances that the mutations can occur. It has a high mutation rate because of a low fidelity viral polymerase it has. The rate of mutation we always specify as 1 to into 10 to the power minus 6 to 2 into 10 to the power minus 6 is the rate of mutation per nucleotide per replication cycle. Although this mutation looks high, but it is much lesser than the other RNA viruses like hepatitis C virus and HIV, which also have a error prone proofreading capacity. So because of that also. So usually the mutations are occurring because of a large genome. There is a replication error which is happening in this virus. It also has a high recombination frequency, as I told you, that it coexists in the animals also, other species also. So the recombinant virus formation is also much higher. 25% of the entire genome can actually cause recombination. So what are the types of mutations we normally see in this virus? One is because of the replication errors, as I told you, that it cannot, so their insertion, deletions keep on occurring. Then sometime in SARS-CoV-2, the host-mediated editing is also there because they are producing errors. So host enzymes are also there to check do the editing. And they are like ApoBec and ADR enzymes, and they also sometimes induce mutations. Then the recombination, as I said, the classical example is the Omicron recombinant XC variant, which formed because of recombination between BA.1 and BA.2. Now, mutations can be called as silent or synonymous, or missense or non-synonymous. The silent mutations are when they occur in a genetic region, but they're not bringing out any change in the protein it is producing. So there is no effect on the structure or function. But missense or nonsense mutations are non-synonymous are those where it alters the protein structure and function. So they are the important one, because once they alter the protein function, the entire the functioning of the virus is affected, and its pathogenicity, virulence, as I said, all gets affected. There are certain nonsense mutations all which bring premature ending to a protein formation, and they lead to incomplete formation of the protein. Classically, the SARS-CoV-2 over the years has shown a lot of acquisition of non-synonymous mutations occurring on the protein structure, which I have told you, 4 important proteins. Next slide. Next slide. So these are the important, so previous slide. So the previous slide, please. Previous slide. So the important notable mutations are the first, so this Wuhan virus, which started in December 2019, the first major mutation occurred in the spike protein region, which we call it D614G. So we always say that aspartic acid, it replaced the glycine. Now this mutation actually made the spike protein very, very stable, improved the entry of the virus into the cells which were having the ACE receptor. So the whole scenario in April 2020, I think this mutation was introduced that led to the entire, globally, all the parent Wuhan virus got replaced by this variant virus very soon. That was a major mutation. Then there are other mutations like at 501 N501Y position, which led to the formation of alpha, beta, and gamma strains, which occurred on the receptor binding domain. So mutations which occurred on these receptor binding domain, which are these classical mutation at 501, 484, 417 position, they enhance the virus combining to the ACE and thus increase the transmissibility. Also, the receptor binding domain is used by the neutralizing antibodies to neutralize the virus, which is used by the vaccine. So the immune escape mechanism also of the virus, it started having once these strains started forming. Another mutation which came at the NTD, N-terminal domain of the SARS-CoV-2 virus, was a deletion mutation at 6970 position. This actually led to the failure of the S gene target detection, which we call as SGTF, and that led to false negative antigen test or false negative PCR assays if the PCR or antigen test were using the surface gene as a target of detection. And they were called as SGTF mutant. And initially in early 2020, these PCR's assays could not pick up the detection of the virus. But later on, the diagnostic companies changed that they made, instead of one, they removed the S gene, and most of the PCRs used E gene and RDRP gene for the detection. Furin cleavage site mutations that led to the high virulence of Delta was P681R2H. That caused high, as I already explained you, if the mutation in the site occurs, it will be better processed. And that is why Delta of all the mutants was processed very fast. The internalization of the virus was fast. Upper respiratory to lower respiratory tract infections and pneumonia and high virulence was there. Its E protein also showed a lot of mutations, because of which the lot of viroporins were released, and the huge cytokine inflammatory storm which we saw in Delta was all because of these mutations. Mutations occurred in the nucleocapsid region also over the year, which led to the increased viral replication and stability in envelope protein as well as the M protein. There is a huge list of these mutations. These are the few classical mutations because of which these viruses evolved. Next slide. So what happens now once a parent virus infects? There are two things, and there are three hypotheses. Now, a person gets infected with the parent virus. When the viral load is very high, it changes because of it acquires a lot of mutations. But at that time, when the high viral load is there, a person transmits this infection to the other person. The chances of passing a variant virus is also very high. But if the infection in the patient is controlled and the viral load is reduced, the chances of transmission of these mutations become less. But if any variant becomes more fit to survive, it acquires, then it becomes the dominant in that patient and can be easily transmitted. What are the three theories how which these variants are transmitted? The first one is, as I said, that a person acquires the infection by one virus, undergoes mutations, and that mutant virus initially is not detected, but once it becomes the dominant virus in a patient population, it starts getting transmitted from one person to the other, and then it becomes detected and it becomes a variant under monitoring and then concern. This is what is happening. There is a silent phase for a long time, and then sudden eruption of a variant under monitoring, which becomes variant of concern also. Second hypothesis is that there is a spillover event occurs from an initial virus. It goes to the animal strains. There it acquires mutation, and then spill back from the animals to humans occur. And then suddenly the spillback virus is a mutated virus, which becomes a variant of concern and start causing more enhanced infection. Third theory is in those patients where a long-standing or a chronic COVID infection occurs. Patients like those who are on immunosuppression and those who cannot clear the virus clearly. In them, what happens is that the virus persists for a longer time, acquires a lot of mutation, and once the infection is transmitted from them to the other patients, it becomes of a variant virus and can cause sudden rise and emergence of any variant of concern. Next slide. So the transmission also depends upon the host factors and the viral factors. So if a variant virus has got accumulated in a person who has got a long-standing infection, at the time of the high viral load only it can be transmitted. It also gets transmitted if it is present at the favoring sites. Means you should have it in your respiratory tract. There should be infection promoting symptoms like cough, sneeze, increased mucus secretion, then only you transmit. The virus promoting factors are that virus has undergone mutation, which has optimized its ACE2 binding capacity. It is better cleaved at the spike protein S1, S2 junction. And on the other hand, it can also evade the immunity a person has mounted. So immune escape is also there. Sometimes change in tissue tropism, like what we saw in Delta. This from the upper respiratory tract, it suddenly became a lower respiratory tract pathogen, causing a lot of virulence and pneumonia. And now, over the year, again, a change in tissue tropism has been noted with the Omicron, that it has again become more of an upper respiratory tract and less of a lower respiratory tract. So all these favors the stages of the viral transmission from the infectious host. Then the mutations can favor the virus to survive outside the host, and then it can establish itself in a new host. So this is how a variant virus travels and during the transmission cycle. Next slide. Now, there are many naming systems which have been adopted for naming these variants. The WHO has named them by the Greek letter, alpha, beta, gamma, delta, and so on and so forth. And they also classified them depending upon their impact on transmissibility and disease causing, like variant of concern, variant of interest, and variant under monitoring, and the risk level they have. Then there is, usually you see the numbers which are written like B.117. This is based on the Pango lineage system. So this is a phylogenetically assigned naming of the global outbreaks number system. And this is how we classify them: B.1, B.11, depending upon the variance it is acquired. And when it becomes more than 5, like B.111, it becomes 5, then we give it a different. So it becomes BA then BA1, 2, and so on and so forth. Alpha, beta, gamma, delta are the ways the WHO is classified. Then there is a next strain system, which was also included the year of identification, like 20A, 19A, B strains, and so on. Mostly we use the WHO alpha, beta, gamma along with the Pango lineage system, by which you have these alphabets and the point numbers given to it. Now, what are variant of concerns? Those which have proven the impact on transmissibility, severity, and the countermeasures. They have a high risk level associated. Till now, these are alpha, beta, gamma, delta, and Omicron, which have been raised as a variant of concern. Other sublineage, all the sublineages of Omicron has also been called as variant of concern so far. Variant of interest are those, means they have the impact, suspected impact, but it is not proven, and suspect community spread is possible. While variant under monitoring are those where the genetic changes are seen, but you don't know how it is going to impact in terms of transmission, severity, or spread. And the recent variant, that is NB.1.8.1, which is causing these infections in India and globally, is classified as variant under monitoring. It has still not been caused as variant of concern. Next slide. So this is what I told you, that these red ones are the variant of concerns which have come up. Initially alpha, beta, and delta, which we all know. And from gamma, the Omicron, then from November 2021, it has been the Omicron variant of concerns which has been spreading, and its sublineages are spread, and it is the one which is continued as a variant of concern and is causing these infections. Next slide. So this is a snapshot of what all changes has occurred. So you have, as from 2021 onwards, the Omicron, it has got 5 subvariants: BA.1, 2. These are the way we name them. Initially, it was BA.1, for BA.2, BA.5, and its subvariants in the form of XBB and JN.1. 2024 end, it was JN.1, which predominated the entire global countries, including India. Next slide. So what has made this Omicron survive so long? So what is happening to this COVID? The Omicron has shown extensive mutations in the spike protein, more than 30. Now, when you have extensive mutations, your capacity to transmit is higher, because we know that it is the one which binds to the ACE receptors. So it has binding affinity, it can cause infectivity, it can evade, there's immune evasion, and it is becoming fitter. The variants have become fitter and fitter. So transmissibility is also high. But the important thing is that its severity is reduced. So virus also wants to survive in the host, in the humans. Now, like the Delta virus, if it becomes highly virulent and pathogenic, it will die out. So virus wants to live in the human beings, and that is why it has taken this milder version. The transmissibility is high, and as I told you, three hypotheses, the variants will keep on coming, jumping from sometimes from animals to humans, or from chronic infections to causing a new variant. But till now, we have only seen reduced severity. Now, because of this reduced severity, it has a silent phase of spread interspersing. So you may have variant of concern rising, but you may have a time or a 1 year, 2 year, where you just have a silent spread of these low severe virus, because this is how the virus wants to survive in the human beings. And this is what is happening with the Omicron. Next slide. Now, this is a very interesting study which was published, which has shown that how globally the mutations have acquired in the four important proteins of the SARS-CoV-2. I just want to highlight in the end that it is highly mutated is the nucleocapsid region, where you can see in across the countries, they have read more than 6.6 million sequences. Nucleocapsid has shown huge amount of mutation, means virus replication capacity is increasing. It can replicate very fast. Then spike protein, huge amount of mutations, and that is why the transmissibility is high, immune evasion is there, and the virus has got the spike protein. The one which is interesting thing is the E protein, which has shown less mutation, almost very less. Now, what E protein does, I told you, it forms those viroporins, those ion channels, and which is responsible for creating a huge cytokine storm and causing an unrest into the host environment. That has not shown a lot of mutation. So because of that, the virus has become less severe. Membrane protein has also not shown much of the mutation which binds. So these are two protein regions which have shown less mutated version over the years. And so they can be a probable future site for a vaccine or therapeutic trials. But this is the snapshot of how the mutations have occurred in the SARS-CoV-2 over the years. Next slide. Next slide. This is our present one publication. We published a lot, but here I want to show, but this is before the Omicron, during the Delta peak, that we also saw a lot of mutations acquiring in all the region. And interestingly, you can see the blue dots are the one, these are the non-synonymous or missense mutation. So most of the mutations which have we also captured from the data we had were of missense or non-synonymous, means they are the one which affect the protein structure and the working of the protein. And that is why the changes in the SARS-CoV-2 occurs, and it affects its virulence and pathogenicity. Next slide. So just summarizing that SARS-CoV-2, although started as an animal virus, got adapted to humans. It has evolved with many mutations, and especially on the structural proteins. Mutations have made it more transmissible, milder, but milder also. Omicron and its subvariants are dominant globally, and it will stay because these mutations are helping the virus also to survive, although it will keep causing up and down in the virus transmissibility and causing these variant of concerns to rise. But right now, it is very mild kind of a virus which is circulating. Thank you. Last slide. So I thank you.
Thank you, Dr. Ekta. An excellent overview. I hope now everybody understands about the virus and how the nomenclature changes.